Greetings, everyone, to the beautiful Oxford Museum of Natural History. It's actually a lovely sunny day here at Oxford, something that does not happen very often, so we should make the most of it. So greetings again to this session number 4, I believe, of the 64th annual meeting of the Palaeontological Association. We're very happy to welcome everyone from across the world. This session is based on flash talks, so 5-minute talks, and at the end of them, at the end of the 5 flash talks, we'll have a joint questions and answers. So please keep sending us all your questions through the chat, and please mark the questions as such using the red circles that will appear at the right of wherever you are typing. Okay, so also a reminder, should you experience any connection issues, please click on the reconnect button at the top of your screen. And I believe those are all the announcements. We have some extra announcements. Remember that, of course, we also have our Discord server online throughout all the duration of the Congress, in which you can interact with peers there, perhaps to talk about research, but also, why not, to catch up. We also have some open rooms for that. All right, it's 9:01 here, UK time, so we will start with the first flash talk. Our first speaker is Candela Blanco Moreno. Candela? Nice, hello. We have Candela there. Hi, how are you? All right, so now I will put your presentation and over there. All right, and it's all yours, Candela.
Thank you. Hello, I'm Candela Blanco from Universidad Autónoma de Madrid in Spain, and I'm going to talk to you about some Heterophyllous ferns from Spain, from 2 different localities. The localities are Las Hoyas and el Montsec. They are both Upper Bryne from the Lower Cretaceous, and both of them are preserved in laminated limestones. So we find these 2 Heterophyllous ferns called Goniopteris laciniata and Sphenopteris wanakoti. Both of them have sphenopteroid pinnals at the bottom of the pinna, as you can see in the silhouettes on the right, and they turn into filiform pinnals at the top. So looking at them closely, we saw that really the morphologies are really similar, the sizes are the same, the transition is the same, so we think it's only one species. For now, we've situated it in Goniopteris laciniata, which is the first one that was described, and we need to do a bit more work to see what the real assignation is. There are 2 different hypotheses for this heterophyly. The first one is that the filiform pinnals are skeletonized because they bear fertile organs. And the second option is that these filiform pinnals developed under the water. So in order to test this and make sure that we have the same plant, we did some architecture analysis using the branching algorithms method, which uses the distance between pinna, dBp in the image, the first segment length, FSL on the image, and the insertion angle of each pinna. So using these measurements, we saw that actually the filiform and sphenopteroid pinnals were different. Architecturally, you can see the green and the blue dots and stars, but that specimens from Las Hoyas and from el Montsec were the same. They are the filled-in and empty circles and stars. So what we saw is that they are indeed architecturally different. We know that normally when there is a differentiation between fertile and sterile pinnals, the architecture is maintained. Only the lamina disappears. In the case of plants that have some leaves that develop underwater, such as ranunculus, the architecture is actually different. So we suspect that maybe the hypothesis of development underwater may be the right one. We also have some evidence on the taphonomy. The brownish compression is the most frequent preservation in this case. We also have a lot of rhizomes. In these localities, ferns are normally preserved as charred remains or impressions. They come from further away. I didn't say this, but these are continental freshwater localities. So the ferns will be living further away from the water. And in this case, we do find rhizomes. We find these brownish compressions that suggest that there is not a lot of transport. So this also indicates that the plants lived close to the water, and this possibility of the tips of some of the leaves developing under the water is possible. Also, in the title, I say they are important in the evolution of vegetation in the Cretaceous. Why is this? This is because it might be one of the earliest evidences of angiosperms and polypodiales. We still have to work on the phylogenetic relationships of this plant, but it may belong to polypodiales. So it would be one of the first moments where we can see that these plants are living in the same environment. So this paper, Ferns Diversified in the Shadow of Angiosperms by Schneider et al., says that polypodiales could have diversified at the same time as angiosperms. We would have one of the first evidences of these plants living together. So this is it. If you have any questions, I will be very happy to answer. Sorry.
Nice. Thanks so much, Candela, for that lovely flash talk. So please keep dropping all your questions for Candela and the remaining of the speakers today in the chat, and we will have an questions and answers part at the end of all the flash talks. All right, so we are on time, so we move on. Our next speaker is Edwin Rodriguez. Edwin, do we have Edwin?
Hello.
Hi there. Hi. How are you? Well,
I'm good. Yeah, I'm good. I just
well, it'll be better as the day progresses, Edwin. Probably for those who are UK time and earlier. All right. So your presentation should be appearing soon, and take it away.
All right. Thank you. Hi everyone. Thanks for joining this talk. I'm Edwin Rodriguez, and I'll be presenting the results of my master project focused on fossil microbes from the Rhynie chert. We have all heard about this iconic fossiliferous hot spring chert deposit that contains superb permineralized fossils of early land plants, fungi, arthropods, roundworms, but also a microbiota that is overlooked. There are fossil bacterial colonies, for example, with well-preserved cell walls and also some internal contents. So the Rhynie chert therefore offers a remarkable and possible unique opportunity on an early terrestrial Devonian microbiological environment to investigate. So the goal of my master project was to begin to investigate these microbial and bacterial fossils on this deposit, but especially to explore how the microbial community and the cellular and molecular information contained in these fossils varies across different microenvironments. For this work, I used petrographic thin sections provided by the University of Aberdeen and on loan from the Oxford University Museum of Natural History. So here I'll present some of my remarks from this work. I have found diverse microfossils, both bacteria and eukaryotes. For example, fungi and amoeba. In many cases, these microorganisms have been already described, but we have found some possible new taxa as well, and we are planning to write it up. Here on the top, we got a cacoi colony-forming bacterium-like organism, and on the bottom left, we have conidiospore-like structures from a possible fungal origin. So this is these are incredible fossils. But I'll come back to this particular two in a moment. Many studies have demonstrated that the ancient carbonaceous matter can contain biosignatures identifiable with Raman spectroscopy. So we applied this method in our samples, and we identified the spectral Raman bands based on the vibrational bonds in carbonaceous material. The D and G bands were very clear, related to disordered and well-organized carbon. And also, this Raman ratio using these two bands is a widely used geothermometer related to thermal moderation. Also, we use the FTIR spectroscopy to identify specific absorbances in specific organic molecular groups. So we obtain the ratio of the CH3 and CH2 chemical moieties that is telling us about the composition of long-chain aliphatic hydrocarbons. Previous studies have suggested that by combining the Raman D and G ratio with the FTIR CH3 CH2 ratio, it's possible to have a chemospace in which we could distinguish between prokaryotes and eukaryotes based on the chemofossil record. Here on the left, we have a plot by Bonneville et al. Based on this idea. However, our data from the Rhynie chert, superimposed in bolder colors, does not fall out according to this method. So we got, for example, a Rhynie bacterium that is plotting as a eukaryote. We got a fungi plotting all the way out from the eukaryotic hole, and all the ratios were similar in values. So this doesn't quite seem to be working. However, by using Raman data combined with the principal component, with a principal components analysis, we might still be possible to differentiate between eukaryotes and eukaryotes. The principal component analysis is suggesting that the Raman biosignatures are capturing information related to biological origin, possibly related with biopolymers of thermal moderation, but this needs more investigation and further work as well. So in conclusion, this work might still have more confirmatory data to obtain and to work, and also to look further for more for a broader data set in order to corroborate all our information. And this is all for me, and thank you so much. Yeah, I'll be happy to answer if there's any question.
Great. Thanks so much, Edwin. Much appreciated. And we'd like to thank the speakers so far for their discipline time-wise. It's much appreciated. We acknowledge the difficulty of cramping everything together, so thank you. All right, so we'll move on now to the next speaker. And please keep typing questions in the chat for all of them. All right, so our next speaker is Emma Dunne. Hi, Emma. How are you?
Hi.
All right, so we'll start your presentation. And here it is. So yours, thanks.
Good morning, everyone. Thank you so much for tuning in. I'm going to give an extremely brief overview of some work that I've been doing with Nussaibah Raja at FAU Erlangen Nuremberg on the trends in Myanmar amber research publications. Myanmar amber has recently received considerable attention, not only because of the exquisite fossils that it can contain, but because of the reported links between the amber mining and the humanitarian crisis currently occurring in the state of Kachin in the north of the country. Over the last 30 years, the number of publications in Myanmar amber has been increasing, with a sharp increase within the last decade. This increase coincides with the takeover of the amber mines in 2017 by the military, who have an economic interest in the mines. Multiple UN reports document the distressing human rights abuses that have occurred since this takeover. Around the same time, we also see the publications of some very high-profile vertebrate specimens in amber, many of which are reflected in Google search data, indicating how they're of interest to the general public. There is also a peak here that may coincide with the publication of a widely read science article, unfortunately marked here as a New York Times article, exposing the ethical minefield of working with this material. Using data that we have been assembling from the paleobiology database for a wider project on the human factors that bias global fossil data, we looked at the affiliations of researchers who are publishing on fossils from Myanmar. Now, Myanmar is not only known for its fossils in amber, but other fossils too, particularly Cenozoic primates. These non-amber fossils are most often worked on by researchers in the global north, that is primarily North America and Europe, as well as China. But there are also some local collaborators from Myanmar on these publications, as seen here on the map. However, fossils in amber are also published on by researchers based in the global north and China primarily, but the key difference here is that there is no local researchers from Myanmar on these papers. This may be due to differences in Myanmar's laws, where fossils are considered antique objects and are illegal to export without a permit, likely fueling the need for local collaborators. However, amber is considered a gemstone, regardless of whether it contains a fossil or not, allowing loopholes in these laws to be exploited. No matter the cause of the pattern, it is still indicative of the deleterious practice known as parachute science, whereby paleontologists, typically from the global north, like me, drop in on another country or area, typically in the global south, like Myanmar, to take whatever is needed to further research without contributing in any way to the local community. Looking forward, the first step is to acknowledge what is happening, informed by the data, and I'm only here to present this data. Many possible solutions have already been proposed by others. Earlier this year, the Society of Paleontology released a letter urging journal editors to boycott submissions relating to amber specimens acquired from Myanmar after June 2017. What this is essentially urging is that each publication be accompanied by a robust ethical statement and permits, and it surprised me that this is not already a common practice. When we looked at a selection of papers on Myanmar amber recorded in the Paleobiology database that were published since 2017, we found only a single paper that made any reference to an export permit. And this is less than the number of Myanmar amber papers that were dedicated to a celebrity, one of these being Mick Jagger of the Rolling Stones on his 75th birthday. But of course, ethical statements alone will not bring about much needed change, and the popularity of Myanmar amber is already soaring, and the issues associated with the material are already becoming more apparent. We need a holistic approach where we work together with the many stakeholders involved in these practices, for example, those who support us, fund us, and publish our papers, as well as begin to work more with local experts from Myanmar, as already done by some non-amber researchers. And finally, as individuals, we must make ethical choices about the work that we do, the collaborative networks we partake in, the information we pass to our students, and the journals we submit to, review for, and cite. I especially call on those who are in more senior positions to use your power and privilege to challenge these unethical and harmful practices and help those of us who are seeking to bring about meaningful changes. Thank you so much again for tuning in. We'd be delighted to hear from you if you have any comments or questions. And that's it from me.
Thanks so much, Emma, for tackling such important matters in science. Please, all the questions that you have about Emma's talk, type them in the chat, and we will tackle them at the end of the session in a joint questions and answers. Thanks so much, Emma, again. All right, so we move on to the next talk. All right, and here's your presentation. And all right, take it away. All yours.
Okay. Good morning, Europe. I have to be really brief. Every time we find an exceptionally preserved fossil in the section, it doesn't necessarily mean that this is the time of the evolutionary origin of the clade. Every time an exceptionally preserved fossil disappears from the section, it doesn't mean that this is an extinction. However, this is not the case with the Ediacaran fossils. The first time they appear in the section is usually regarded as the evolutionary origin, related to an increase in oxygenation of the ocean or the atmosphere. And when Ediacaran fossils disappear from the section, this is usually regarded as a mass extinctions, and Bilaterians are to blame. However, there is growing evidence that Ediacaran organisms were adapted to live in anoxic conditions. In the photographs from left to right, you see holdfast with root-like extensions. Why would the holdfast have root-like extensions if not feeding feeding in the anoxic sediment underneath the microbial mat? Why would Dickinsone and Georgia leave the trackways on the microbial mats if not for feeding anoxic sediment underneath the mat? Why would Nama-type organisms go underground if they were not adapted to live in anoxic sediment? And recent interesting analysis of the distribution of iron species in the fossils from Caddispeed. They clearly show that all these organisms were adapted to live in an oxic environment. And the diagram in the right-hand side of this slide, it's a working hypothesis where you see the orange color stands for anoxic environment and blue color stands for the oxic environment. And this is new data, new unpublished data from an Ediacaran section from the Arctic Siberia, where you can see where the results of the study of uranium isotopes by J. Kaufman and biomarkers by colleagues in my institute. They demonstrated that the entire Caddispeed formation was deposited in an oxic environment. The red color stands for an anoxic and possibly euxinic waters, and green and blue, it's based on biomarkers. They stand for still anoxic, but without euxinia. And as you can see, the fossils that are preserved as casts and molds in the right-hand side of the slide, they are found in the parts of the section where the conditions slightly change from euxinic, non-euxinic. And in between these intervals, the fossils are shown in the left-hand side of the slide. You see the fossils preserved not as casts and molds, but as faintly visible outlines with no relief. So far we found only 5 specimens, and they're all shown on these slides. And there is Charnia, there is Holdfast with root-like extensions, and there is front. They're preserved not as casts and molds, whereas the fossils shown to the right are the same taxa, but preserved as casts and molds. And all I'm trying to say is that the entire Caddispeed formation is represented by the same type of Ediacaran fossils. However, they are preserved only in those parts of the section that are marked by sudden changes in the redox environment. So a sudden shift in redox conditions probably facilitated early diagenetic cementation of the sediment, and this promoted the preservation of Ediacaran fossils as casts and molds. Whereas in the part of the section where there were no shifts in the redox conditions, the fossils are not preserved, but they are there. We know this from our detailed studies of the section, and we're just lucky. We're just lucky with this section where we were able to show that the fossils do not, the organisms, they don't go extinct. They just disappear for taphonomic reasons. And I'm pretty sure this is the case in other localities, and we should be very, very careful with interpreting the time of the origin of eumetazoans and the time when they went extinct. And I would say that they possibly never went extinct. It's just the closure of the taphonomic window because of the stabilization of the redox environment. So probably I should stop here. Thank you very much.
Thank you so much, Dimitri. That was really interesting. And here we've now reached the last flash talk of today. So I will now invite all the speakers to turn their cameras and microphones on. Actually, we have a bit of a discussion there. You can hear some noise in the background, so probably if you guys have some noise around, it would be better if you mute yourselves. I don't know, would you mind switching your microphone off and then we only turn it on in case you have anything to say. Feel free to engage in discussions, like I said. So, all right. So some of the questions in the chat. First one for Edwin. For instance, I don't believe you have answered this one. How do you take into account the very different thermal maturity levels of your samples?
Yeah, actually, I was typing out that answer. So we use the Raman spectra. What I did was following the principle by Kouketsu from 2014. They used a geothermometer based on the convolution of the Raman spectra. So by checking the different intensities ratios that you are having, and later applying this data on PicFig software, you are able to have a deconvolved spectrum. And later taking on account the full width half maximum values, you can correlate that in order to place later on all the samples in low, medium, or high metamorphic grade temperature maturity. Yeah.
Thank you. All right. One question in the chat that popped up for Emma. Do we see similar issues surrounding other amber deposits with respect to their specific geopolitical setting and issues?
Oh, great question. So the three main sources of amber that get talked about in the literature are Myanmar, Baltic amber, and Dominican Republic amber. So Myanmar amber has been given the most attention, of course, with respect to ethical concerns. But there is reports of environmental problems with the way that Baltic amber is being mined. And there are some uncertainties as to how Dominican amber is sourced with regards to human rights and stuff. But I suppose I will caveat all that with saying that I'm not actually sure, but that to bear in mind that when things are being mined for commercial interests, that we do need to be mindful of why that's being done and how that is being done.
Exactly, and I guess also when those samples hit the online market as well, right?
Exactly.
Yeah, definitely. All right, so other questions? Dima, one question for Dimitri here. One for provoking our thoughts. What metabolic pathways do you envisage for example, the use of sulfur in sediment plus oxygen from water, something else?
Well, it's really difficult to say. Perhaps they could harbor bacteria. I mean, many organisms, many metazoans that we have today are adapted to live in an oxygen environment, and they use all sorts of pathways to deal with this. It's all entirely, completely hypothetical. You know, we can play with all sorts of ideas. Really don't know.
And another question for you here. We'll see. Dima, do you think that the taphonomic window didn't exist further back in time? So perhaps we could have white sea biodiversity back before 565 million years ago.
Yes, it's a reasonable thought. The Ediacaran period is very interesting because of the oscillation of the redox conditions, and this is when all these taphonomic windows they start opening and closing in the shallow marine environment. I would predict that if we look carefully, we will eventually find these fossils further down in the section.
Who else have we got? Nice. And one question more for you: is there any reason why the fossils are preserved in higher relief in the rapidly changing redox zones?
Yes, I think it's because of the rapidly changing redox conditions. They promote early diagenetic lithification of the sediment prior to the compaction. So the fossils are preserved as concretions, whereas the sediment around them was not lithified, and that's why they as a result of the compaction they produce this high relief.
All right, so no questions, Candela, Lara, but there was actually a lot of excitement about your talk in the chat. So I'm sure that people will contact you directly in case they want to share impressions with you. So please keep using all the means that we have to interact with one another in these difficult, times in which communication is more important than ever. We have our Discord server. Please join everyone there for more discussions. And thank you so much to all the speakers for their much appreciated discipline with time-wise and for the wonderful contributions. It's not easy to cramp everything together into 5 minutes, so thank you so much to all. Thanks so much to all the people in the chat, all of the attendees, also for asking very good questions and redacted in a very easy way to convey, so that's also appreciated. And the next session will start in about 5 minutes. Remember that it's session number 5, also flash talks. And remember that in order to get access to that session, you need to click to a new link that you will have received. So yeah, in 5 minutes, that session will start. That's 9:40 UK time. So see you all there. Thanks so much, everyone.